Hi, I'm Mr. Baker, and this is Unit 9 Protein Synthesis, 9.3 and 9.4, which is Translation, Genetic Code, and Mutations. Translation is the second step of gene expression, where we're going to take a segment of mRNA and translate it into an amino acid chain, which is going to be polypeptide or a protein, and it's going to be done by the ribosomes. This amino acid chain is going to fold to form proteins, and it occurs on the rough ER in the cytoplasm. There's a video that you should have watched. And here are the ribosomes. Ribosomes moving down the mRNA, and it is churning out amino acids. Translation, there's another little GIF for you. You need to remember that each amino acid has a specific set of 3 base pairs, or a triplet, that codes for it, and that's called a codon. There's the old box, but we've been using the codon wheel. How to read the codon chart, we've covered that in class, and here's a video to help you that you've watched as well. Realize that codons are 3 bases that code for an amino acid. There are 64 different codons. 61 of those code for amino acids. There are 4 that I ask you to memorize. AUG is the start codon, and that codes for an amino acid called methionine. UAA, UAG, UGA are the stop codons. Translation is 3 steps: initiation, elongation, and termination. Another video that you should have been assigned. What happens during initiation is mRNA attaches to the smallest of the ribosome subunit. There are 2, a small and a big one. Methionine bound on the tRNA binds to the start codon on mRNA, and the large ribosome subunit binds to make what we call a sandwich. And there's a picture. There's your small piece is the small brown piece. Your mRNA is the red. Your tRNA is the bent firecracker with methionine on the top, because that is the start codon. Then the big piece comes down and fits in. Elongation is with tRNA keeps coming in, and the ribosome slides down the line, adding amino acids, and they keep transferring over and over and over until you get to a stop codon. That is termination. The stop codon attracts something that we're going to call a releasing factor that wedges the whole business apart. And this releasing factor is this yellow dude who comes in and just kind of forces everybody apart. The big idea, the DNA, the nucleus, is transcribed into a single-stranded molecule that is mRNA. mRNA travels to the ribosomes, which is in the cytoplasm or attached to the rough ER. This mRNA is then translated into an amino acid chain. Don't forget that ribosomes, which are made of rRNA and transfer RNA, tRNA, help this whole process. This amino acid chain then folds into a 3-dimensional protein. And this protein is going to do work, provide structure for the cell, maybe it's an enzyme. There are about 20,000 genes in a human. Is every gene turned on at every moment? Of course not. So it stands to reason that various genes are turned on at various times in different cells as you need. For example, you don't need a molecule in the retina to be working in your liver. So you turn it on in the eye and you turn it off in the liver. The evolution of it, which came first, DNA or RNA? RNA came first. It's able to do some job on its own, but it mutates real fast. So DNA evolved as a master copy, as it doesn't mutate as easily as RNA. And think of DNA as your cloud backup of a commonly used file. There was a website I wanted you to go to and play with. Here is a table that you should have had a quiz on. And that's your quiz, 9.3. Mutations is our next one. These are changes to the DNA or pieces of chromosomes. A mutation is any change in the DNA code that causes a change in the mRNA, which then will cause a change in the amino acid order. Point mutations can arise from spontaneous mutations. Those are things that happen without planning, and sometimes that occurs during DNA replication. It makes a mistake. Things that accelerate this process are called mutation generators or mutagens. The big 4 are UV rays, X-rays, incredibly warm heat, or various chemicals. Gene mutations. Now, these are mutations in a specific gene, a small piece of DNA, and they include point versus frame shift. A point is where you're going to change one base to another. You change a letter. It's also called a substitution. A frame shift is when you take one out or add a new one in and it shifts everything downstream of it. Hence, frame shift. If you add one in, it's called addition. If you remove a base, it's called a deletion. Here are some various diagrams that help. Here's your gene mutations, where you've got insertion, deletion, and substitution. So let's talk about mutations in light of the pandemic. All this information is current as of the end of February of 2021. SARS-CoV-2 has a spike protein which binds to the human ACE2 receptor. This ACE2 receptor is expressed in tissues all throughout the human body. The spike protein has two pieces to it, S1 and S2. S1 binds to the ACE2 receptor. S2 helps bind to the host cell membrane. You've got to have S1 to attach, S2 to fuse. This S protein is 1,273 amino acids long. Due to the fact that SARS-CoV-2 is an RNA virus, it will change its genetic code or mutate readily because RNA polymerase isn't perfect when making copies of RNA. The first major variant or mutation that became noticed was the D614G, which became the dominant strain worldwide by the summer of 2020. So how do you read this? D is the original amino acid. 614 is the position out of 1,273, and G is the new mutation. So D was ASP, and the original amino acid, 614, is where it was located out of the 1,273 chain, and G is glycine, which is the new mutation. There are other variants. E484K is one of concern. K471N is another one, and this N501Y, which is the first seen in the UK and South Africa, which is now showing up everywhere. These three mutations occur in the receptor binding domain, which means it makes it better able to bind to the ACE2 receptor, which makes it more infectious. Here is a nice diagram of the N501Y mutation and how it binds to the ACE2 receptor, which is up in green. These are all point mutations or substitutions specifically. And what's happening is not so much that humans are traveling around and spreading the virus, is that the virus is showing what's called convergent evolution, where these mutations occur, and because they're advantageous to the virus, they are retained. It gives them an advantage. And it's occurring in different parts of the world independently of each other. So just because it was first identified in South Africa or the UK doesn't mean it's not going to occur here as well. These three mutations raise concerns that the S protein has changed its shape enough to evade the memory cells from the earlier variant, the D614G, meaning, and we know this, that you can be reinfected with the new variants because it's completely new to your immune system. Evidence so far suggests that it is not escaping the vaccine. The vaccine gives a stronger immune response. So far, so good. We do need more information to make sure that we are all protected and that these new variants do not allow themselves to be transmitted by people who are vaccinated. So more information, perhaps I'll update this next year. There's where I got my information. Let's talk more about mutations. And we need to talk about entire chromosomes being mutated. These are all due to errors in crossing over. Remember prophase 1 during meiosis? Well, when it happens right, you increase variation. That's a good thing. But sometimes it doesn't happen right. For example, you can try to cross over and lose one of those pieces, and that's called a deletion. Sometimes it's supposed to transfer to a different one, and it reattaches after it's already crossed over, and that's called an insertion or duplication. You could also switch with somebody else you're not supposed to switch with. That's called a translocation. Trans meaning different, location, obvious, different location. And sometimes you get flipped upside down and reattach, and that's called an inversion. Again, these are all issues in crossing over. There's your deletion, your inversion upside down, translocation to a different one. Duplication is when you get the same piece. Deletion is when it disappears. That's for your quiz 9.4. Study well, and I will see you next time.